When we say antibodies, now antibodies, they might be either monoclonal, or they might be polyclonal antibodies. Fine. Now this monoclonal antibodies, they are what kind of antibodies are they? Those actually, we know that antibodies are the glycoprotein that are synthesized in the blood. Yeah, and they are synthesized why? To fight against specific antigens, to combat against specific antigens, and so that our body gets the immunity to fight against them. And these antibodies, they are heterogeneous and they are polyclonal antibodies. Now the word poly means many. That means this kinds of, there are certain kinds of antibodies which are not specific for one kind of antigen. They are just like all purpose types of antibody. Okay, they are used in general for combating many kinds of common diseases. So those are known as polyclonal antibodies, that are and those kind of antibodies are produced in our body. Now uh, sometimes what happens, there are certain specific lymphocytes. Okay, those lymphocytes, they can be isolated and then they can be cultured in vitro. In vitro means outside the body. Outside the body, human's body, they can be cultured. Those antibodies can be prepared and they are made capable of producing only one single type of antibody. Now that single type of antibody will have some specificity. No, they will be specific against only specific types of antigen. They are not used in general for all kinds of antigen. They will be specific towards all, certain kinds of antigen, and those kinds of antibodies which are specific, they are known as monoclonal antibodies. So actually, what we are going to learn, one topic, what I was telling you all is the production of monoclonal antibodies. How is this monoclonal antibodies produced? That's what you are going to learn. But prior to that, before learning monoclonal antibodies, there are certain types. Okay, actually the procedure by which these monoclonal antibodies are produced is known as hybridoma technology. Means the cells are actually fused, and that is the reason different cells are fused. So that is the reason the word hybrid has been used. Hybrid is like when two different things join, that is known as hybrid. So that's the reason why this hybridoma, this technique is called the hybridoma technology. The term hybridoma is used to fuse cells, resulting due to fusion of two types of cells. Okay, what are the two types of cells that are fused? They are lymphocytes and tumor cells. The lymphocyte will be that particular cell which are actually helping in producing the antibody. Those are the lymphocytes, and the tumor cells are the cells that those cells are used. Why? Because we know that tumor cell have characteristics of growing for like infinite times. They can grow very fast, and they can produce several cells at a very fast speed. So that is the reason why these tumor cells are used, which are known as myeloma cells. And since these two different types of cells are used and they are fused, or a hybrid is produced, that is why this technique is known as hybridoma technology, which is used for the production of monoclonal antibodies. Okay, so before we actually go into the whole technique of studying the hybridoma technology for production of monoclonal antibodies, we will study on few terms which I'll be showing you all with the help of some video, and then that will clarify uh, certain points that will help you to understand the hybridoma technology.
Let's have a quick overview of antibody production by B lymphocytes. We have studied earlier that origin and maturation of B cells occur in the bone marrow. Then these mature, naive B cells are released into the blood. They keep recirculating between lymph, blood and secondary lymphoid tissues. These B cells have specific receptors on their membrane for antigen recognition. These receptors are known as B cell receptors. Recall that B cell receptors are membrane-bound immunoglobulins. Their structure is similar to that of antibodies. Every day about 1 billion B cells are produced in our body. The B cell receptors present of these cells have pre-existing specificities. Therefore, B cells are able to recognize and react to any possible antigen which it encounters. Each B cell can display about 1 lakh B cell receptors on its surface. Important point here is that all these B cell receptors are specific for only one particular epitope on an antigen. When a mature B cell encounters an antigen, it gets activated and proliferate into a large clone of B cells. Some of them differentiate into antibody-producing plasma cells, and others become long-lived memory cells. Both these plasma cells and memory cells will be specific to the antigen or more specifically the epitope, which the mature B cell encountered initially. Now that we are clear how antibody production takes place, let's come to the topic of today's video lecture: polyclonal antibodies. Suppose this is an antigen which invaded the body. Let's say there is some bacterial infection. We know that microbial surface has many molecules that vary in their chemical nature. They may be proteins, carbohydrates, lipids. These microbial surface molecules or their portions are recognized by B cell receptors. These are the epitopes to which B cell receptor will bind. We also said that B cells of diverse specificities are circulating in our body. For our illustration, we will take 4 such B cells. Each of these B cells is specific for a single epitope on the antigen. So when there is an invasion by the antigens, these B cells recognize and bind to their specific epitopes and get activated. Each of these B cells will proliferate and produce large clone of cells. Some of these cells in each case will get differentiated into plasma cell. And each of these plasma cell will secrete antibodies specific to the epitope which triggered their differentiation. So, as a result for this antigen, our immune system produced 4 types of antibodies, each specific for a different epitope on the same antigen. Antibodies derived from multiple B cell clones are known as polyclonal antibodies. Polyclonal antibodies are a heterogeneous mixture of antibodies, each recognizing a different epitope on the same antigen. We now understand that polyclonal antibodies arise from many B cell clones and have a heterogeneous collection of binding sites. Production of polyclonal antibodies in response to pathogens or antigens has a very important advantage in vivo. These antibodies offer multiple ways to attack a pathogen, such as by facilitating phagocytosis, complement-mediated lysis, etc. Antibodies are a great tool in diagnostic immunology. This is because the antigen-antibody interaction is highly specific. An antibody can detect one antigen molecule among 10 to the power 8 other antigen molecules. Antibodies are used to locate and identify antigens, to purify, characterize, and quantitate antigens. Polyclonal antibodies are useful in agglutination and immunoprecipitation-based techniques. This is because these techniques involve large antigen-antibody or immune complex formation. Polyclonal antibodies are efficient in this because they provide multiple ways of binding to the target antigens. Main source of polyclonal antibodies is human or animal blood. For polyclonal antibody production, an organism, for example rabbit, is immunized with the antigen against which we need polyclonal antibodies. Rabbit is injected with the antigen. This is done 1 or more times. Rabbit's immune system will respond to these antigens by producing antibodies. After some time, these antibodies are isolated and extracted from the rabbit's serum. This way we get antiserum containing polyclonal antibodies targeted against the desired antigen. But usage of polyclonal antibodies in diagnostic immunology has some disadvantages. For research and diagnostic usage we require antibodies in large quantity, but in a polyclonal mixture of antibodies, desired antibody is present in low concentration. The polyclonal antibodies may have cross-reactivities. This will reduce their sensitivity and specificity. Moreover, the isolation of desired antibody from a polyclonal mixture is expensive, time-consuming and less efficient. Alternative solution to this was provided by Georges Culler and Cesar Milstein. They developed monoclonal antibodies. These antibodies are homogeneous and all recognize the same epitope. We will talk about monoclonal antibodies and their production in next video lecture.
Monoclonal antibodies, they identify only similar types of epitopes. Okay, now you have learned about polyclonal antibodies. Now before we start the monoclonal antibodies, for the whole process of production of monoclonal antibodies, what are the things that you need to know? You will have to know about the cell fusion. Okay, then you will have to know what is the media that is used for the production of this monoclonal antibodies, because the monoclonal antibodies are not produced within the body. This technology, which is hybridoma technology, it is done outside the body of the host. Okay, so that is why you will have to know the medium and the enzymes that are required for this, for the production of this. Okay, so now we are going to see what is cell fusion and then about the media that is used for the production of these antibodies.
Cell fusion is a technique in which two different types of cells or cells from two different species are fused in vitro. This is done in the presence of chemical fusogens such as polyethylene glycol or by electric shock. Suppose these are two different types of cells, represented by X and Y. These cells are allowed to fuse by exposing them to chemical fusogen polyethylene glycol. The cytoplasms of these two cells combine and form a single large cell with two nuclei. This cell is known as heterokaryon. Hetero means different, and karyon means chromosomes. It is so called because this cell has two different sets of chromosomes. In the next step, this heterokaryon undergoes cell division, and the two nuclei also fuse. This resulting cell now contains a single large nucleus having two sets of chromosomes. This cell is known as somatic cell hybrid. Somatic cell hybrid is unstable. This is because of the presence of the extra set of chromosomes acquired during fusion. These extra chromosomes are lost randomly during subsequent cell divisions until the cell returns to the normal diploid state of one of paired chromosomes.
So this is what is cell fusion. So this particular technique is going to be used during monoclonal antibody production. Okay, so here they have shown two different types of cells. Similarly, while production of monoclonal antibodies, also two different types of cells will be used. One is the lymphocytes, another is the tumor cell. Okay, now these two different cells will be fused to each other, and the fusion of these two cells will be done with the help of uh, this particular thing, which is known as polyethyl glycol (PEG). Okay, and that will after using PEG for this fusion, then later on this steps will be followed in order to produce this somatic cell hybrid.
This means cell increases in number. And this increase in number is the result of cell division. Let's say this is a cell, it undergoes single event of cell division. As a result, two cells are formed. The most important event which happens during cell division is DNA replication. Without this, cell proliferation is impossible. We all know that DNA is made up of nucleotides, namely adenine, guanine, cytosine, and thymine. These nucleotides are synthesized cell. Now suppose a cell is not able to synthesize these nucleotides. What will happen? There will be no DNA formation, hence no DNA replication, and this will be fatal for the cell. Let's have an overview of how these nucleotides are synthesized in mammalian cells. In the mammalian cells, there are two main pathways of the nucleotide synthesis. These are de novo synthesis pathway and the salvage pathway. De novo synthesis pathway. De novo means starting from the beginning. In this pathway, nucleotides formation begins with simple precursor compounds. Through a series of steps, this pathway leads to the formation of nucleotides that finally forms DNA. At the initial step of de novo pathway, tetrahydrofolate is essentially required. Without tetrahydrofolate, this pathway cannot proceed. Tetrahydrofolate is made from dihydrofolate, and this reaction is catalyzed by the enzyme dihydrofolate reductase. If formation of tetrahydrofolate is blocked somehow, synthesis of nucleotides by de novo pathway will automatically fail. One such drug that inhibits formation of tetrahydrofolate is aminopterin. Aminopterin is an enzyme inhibitor. It competes for the folate binding site of the enzyme dihydrofolate reductase and thus blocks tetrahydrofolate synthesis. Overall, we can say that aminopterin blocks the de novo pathway of nucleotide synthesis. Now let's come to the second pathway, the salvage pathway. Term salvage is derived from Latin, which means to save or retrieve. This pathway recycles free bases and nucleosides inside the cell and form nucleotides. In other words, this pathway recovers preformed bases and form nucleotides by reconnecting them. The salvage pathway involves two key enzymes abbreviated as HGPRT and TK. HGPRT stands for hypoxanthine guanine phosphoribosyl transferase. This enzyme catalyzes the formation of nucleotides from hypoxanthine and guanine. Second enzyme TK stands for thymidine kinase. This enzyme converts thymidine into corresponding nucleotide. If there is any mutation in the genes producing either of these two key enzymes, salvage pathway will be blocked. This is because the mutation will make these enzymes non-functional. So, we now understand that HGPRT is a key enzyme in the formation of nucleotides by the salvage pathway.
Now, did you all understand this one? This video which I have shown you right now.
Yes, ma'am.
Okay. Now, in from this video, what is much important to us for the whole process of monoclonal antibodies production is this particular enzyme which is called HGPRT. Okay, so since we are producing this monoclonal antibodies in vitro, we will have to add some certain things into that into that media where the production will be done. So there we will be adding this particular enzyme HGPRT so that the synthesis of nucleotides may be done, and the salvage pathway is followed in that production of monoclonal antibodies. As it is told that salvage pathway is what means it retrieves the old cells which are already present in the cells, and here we are actually transferring the cells from a different animal into the media. So that is why here retrieving of the cells is necessary, and so it will follow the salvage pathway. And when it follows the salvage pathway, HGPRT enzyme is necessary in that, so that there is synthesis of the nucleotides for further production of the cells. Okay, and this is for information that if in the cells of in the like body of any animals or human beings, there is synthesis of nucleotide, the inhibition of the pathway that is follows is de novo pathway, and this de novo pathway can be inhibited by the use of this particular substance called aminopterin. If sometimes it is, if it is necessary to block the pathway, then drugs having the presence of aminopterin is prescribed so that the pathway can be blocked and there is no further production of DNA. Now we don't have much time, we just have 5 minutes left, so we'll stop this here. I have 2 more videos to show you all, so probably in the next class we can do that. Or do you have any class now? What class do you all have now?
EVS.
You have EBS, so sir will take your class. Okay, let me see if in the afternoon, if we get the time, we get some time, I can show you that video. And also we have not done anything in practical, so we'll have to do practicals also, okay, online. So you'll just remind me when you all have practical classes so that I can take few classes for you all. Respond to your roll call.
Ma'am,
Yeah, say.
ma'am, practical মানে practical theory টো হ'ব নেকি online?
অনলাইন থিয়ৰিয়ে হ'ব আৰু কেনেকৈ প্ৰেক্টিকেলটো কৰাম বাৰু।
সেইটোৱে।
দেৱাংশী, সিদ্ধাৰ্থ, মনীষা, পামিতা, Sara,
Present, ma'am.
Priyanka,
Present, ma'am.
Himanshu,
Present, ma'am.
Suman, Tushar, Mona Lisa,
Yes, ma'am.
Kanan, Bitupon, Dipjyoti, Nikita,
Present, ma'am.
Nilpavan, Sayasri, Hirok,
Present, ma'am.
Ma'am, Sayasri present.
Samir, Irani, Barkha,
Present, ma'am.
Suparna, Anjan, Amit, Shreyashi,
Present, ma'am.
David,
Present, ma'am.
Elena, Anjali,
Yes, ma'am.
Sania, Jintumoni, Sanchaita, Suman Paul,
Present, ma'am.
Darshana,
Present, ma'am.
Sapna, Yaljomo,
Present, ma'am. Ma'am, Sapna present.
yes, Indra, Shivaji, Moromi,
Present, ma'am.
Rajashree,
Present, ma'am.
Ritumuni. Okay. Was anybody left out or what? Just go through the screen once. Sapna, I have given.
Ma'am,
I have given your
roll
attendance.
number
Sapna.
35
I have
present.
given your attendance already.
Oh, okay, ma'am.